right, guys, we are going to be getting into protein synthesis. So this is bringing everything together with our DNA, our RNA, and our replication. So if you recall with me real quick, DNA contains the instructions to make all of an organism's proteins. So RNA is what helps DNA make proteins. So this idea is that DNA leads to RNA, which leads to proteins. And we call this the central dogma. So the central dogma explains how life is determined through DNA. So this is very, very important, especially if you want to go further into sciences, if you're going into nursing, anything like that. Your central dogma is going to be very important for you to remember and to understand. So your DNA contains genes. So a section of DNA that codes for a protein is called a gene. The gene is read and the message is used to make a protein. Proteins then determine traits such as eye color or if you have dimples, for example. So your gene is found in your DNA, which is found in your chromosome, which is found in your cells. Now, protein synthesis is the process of making proteins, but there are two steps. You have transcription, which is when your mRNA or your messenger RNA copies your DNA instructions, and then you have translation. Translation is when ribosomes use the mRNA or the messenger RNA to actually make the proteins. So let's talk about step one, transcription. Transcription occurs in the nucleus, and it involves DNA and your mRNA or your messenger RNA. So basically what happens is your DNA is going to unwind itself where the gene is, because that's what we want. So the RNA polymerase uses DNA as a template to make a copy of the mRNA. So it's a transcript, just like what you get from school with all your grades. Now, the mRNA copy can leave the nucleus so we can start our next step. So here is your strand of DNA, your strand of mRNA, and here's your RNA polymerase. So it's going to move through and it's going to add in our complementary base pairs. And remember, we're working with RNA, so we no longer have thymine. It is replaced with uracil. So it's going to come in and make this complementary RNA to go with it. Now, the newly made mRNA is going to leave the nucleus. I also want to refer back to genetic code for a minute. Remember that we are trying to make a protein. Right now, we have an mRNA message. So that's nucleic acid language needs to be translated into protein language. To do that, we need to understand the code. To understand the code, we need our codon chart. So mRNA has codons. So a codon is a sequence of 3 nucleotides that code for an amino acid. And amino acids are the building blocks of our proteins. So the mRNA sequence is going to be segmented into sets of 3 to make one codon. So you can think of codons like the words of the genetic code. You need to translate all the words in order to get the message. And in this case, the message would be protein. Now, this is one of my favorite parts of DNA replication, transcription, translation, everything. This is my absolute favorite part. I love this codon wheel. It's so easy to use and it's so much fun because you are playing a detective to figure out this secret language. So I'll teach you how to use this chart. So let's look at our first sequence here. So our first sequence has a U. Uracil is the first part of our codon. So what we do is we come over here to our circle chart. The innermost circle will represent the first letter in our codon. So in this case, our first one is U. So we find our U. Oh, here we go. Now U represents 1/4 of this circle. So we know we are only going to be looking at these. This is all we're working with here. But that's still a lot to choose from. So let's break this down a little further. Now the second innermost circle is where we need to go next. This is what's going to be our second nucleotide. So our I apologize. So our second one is A. So we know that we're still working with U, that hasn't changed. So we need to find our A. So we don't need that, don't need that. Here we go. So we go UA. So now we have isolated an even smaller section here. So it looks like these are our two choices. But we have to figure out what the last nucleotide is to be able to distinguish whether this is our protein or if we have a stop codon. So let's check and see what our last letter is. Our last letter is C. So we go U, A, and C. So we know that this is going to be our protein. So let's do it with the second codon. So we got a G here. So we know that we are working with this 1/4 of the codon chart because we looked at the G in the innermost circle. Next, we have our C. So we need to find our C. Here we go. Oh, well, look at that. You don't even necessarily have to know what the last nucleotide is because look at that. Anything that starts out with a G and a C, no matter what the last nucleotide is, it's always going to be this protein. So it is very easy, very fun to figure out. So now, let's figure out which amino acids these codons will code for. So you're going to start in the middle and you're going to work your way to the outside. So we got U, we got A, we got C, right? Boom. Normally, so we don't have to write out this whole thing because that can become a lot. So a lot of times scientists will just write the first 3 letters of the protein, which can also work for you. Now let's check out our next one. We've got GCU. Got GCU. Look at that. Boom. You guys are so smart. It's just so easy. Now, remember, we have 3 different types of RNA that all have different jobs. So your messenger RNA or your mRNA is what carries your DNA message as codons. Your rRNA or your ribosomal RNA makes up the ribosomes, while your tRNA, that matches the anticodon to the mRNA codon to bring the correct amino acid. I like to think of the mRNA and the tRNA as the waiter and the cook. All right, you've got the waiter that went over to the table to take your food order, right? So they're going to take that message back to the cook. Like, hey, yeah, I'd like a cheeseburger, a side of fries, and a milkshake. And that transfer RNA is going to take that order, prepare it for you, and bring it out. And that's how it works. It's going to work together like a kitchen staff in a way. So let's talk about step 2, which is translation. Translation is going to occur in the ribosomes. Now, your players that are involved in translation is your mRNA or your messenger RNA, your rRNA, and your tRNA. So here's what happens. Your mRNA finds a ribosome and it's going to bind to it. After it finds that ribosome to bind to, the mRNA codons are going to be read by the tRNA. So the tRNA will bring in the right amino acids that matches the codon. Because just like when you order food in a restaurant, if your food comes out incorrectly, you're not going to be happy about it. Just like your DNA won't be happy if its amino acids are messed up. So the amino acids will then link together to form our proteins. So we have our mRNA sequence here. Oh, look at that. We have a pretty purple blob. What is that pretty purple blob? That is our ribosome. And here, that's where we have our tRNA coming in. Our tRNA brought in this amino acid for us. Now we're going to keep going down. So we've got our codon, and your tRNA brought us this anticodon to bring us this amino acid by reading the codon chart. And it continues all the way down. Now, your two amino acids up here are going to bind together with it's going to create a polypeptide chain. Now, I want you guys to practice with this. So I have given you a DNA sequence. What I'd like for you to do is transcribe this DNA sequence into mRNA, into an amino acid sequence. All right, so first we have TAC. Let's see what our mRNA is. AUG. Because remember, A and T are going to go together. But we don't have T's in mRNA, so our A is going to turn into a U, and our C and G is always going to go together. Next, we have AGT. So that will turn into U, C, A. Okay? Next, you got an A to a U, a T to an A, and another A to a U. All right, we're halfway there. And here are your last three. So now, in case you didn't get it, I have given you the mRNA codons. So now it's your job to take these to read your codon chart. So I'll help you with the first one in case you forgot how to use our chart. So first, we have an AUG. So remember, our first letter is going to associate with the innermost circle of the codon chart. So since our first letter is A, we know we're only going to be working with this 1/4 of our chart. Next, we got a C. Oh, we got a U here. So we need to find our U. All right, here's our U. So now our last letter is going to dictate what amino acid we choose. So it's going to be A, U, G. So here is our amino acid. Boom. So now I want you guys to try the rest on your own. All right, guys, I've given you guys a few seconds, so let's go in and let's practice this together. So we have our second sequence of mRNA. We have UCA. So if we use our codon chart, we're going to go over here. Our innermost circle is going to be our U. So we know that we are going to be in this 1/4 of our codon chart. Next, we have our C. So let's find our U and our C. Well, look at that. We don't even have to figure out what our next nucleotide is because U and C, it all codes for the same amino acid. So there's our second one. All right, we got 4 more to go. Okay, so you have U, A, U. So once again, we are going to be back on this side of the codon chart. We're going to our A, so it looks like we've got two different choices here. So what's our last letter? Okay, our last letter is U. So we know there's our amino acid. All right, let's do another one. So we got C, G, and U. So we know with C, we are going to be on this 1/4 of the codon chart. We are going to go to our G side, and look at that. We don't even have to look at our last nucleotide because we only have one option for an amino acid here. All right, two more. All right, we got our C, which leads to our C. And look at that. Still, once again, we don't even have to look at our last nucleotide. See, I told you guys, this is so much fun. This is my favorite part. Okay, last one. G, A. We got another G. We have our last amino acid. Our amino acids are then going to be joined together with polypeptide bonds, and that is what is going to create our protein. And after an exhausting time of being our investigation people, we are done. So make sure that you guys completed all of your activities that went along with it.